Hello, my name is Julie Luna. Chrysanthemum is the target chemical that this research will be concisely dedicated to. I will first discuss the overall history of the plant. In the 15th century BC, the chrysanthemum was first discussed in writings native to China. China cultivated this plant as a flowering herb. In other words, China was the first country to develop this plant for human use. China thought very highly of this new renowned herb and even believed that it contained the power of life. According to Chinese legend, different parts of the plant held different remedies of cure. For example, boiling the roots helped diminish headaches. Likewise, young sprouts and petals were eaten in salads, and the leaves were brewed for tea. As chrysanthemums began to appear in other parts of the world, so did the fascination with them. Around the 8th century AD, Japan took an extreme liking for the flower and adopted a single flower chrysanthemum as the crest and official seal of the emperor. Japan even developed a national chrysanthemum day, which is called the Festival of Happiness. By the 17th century, chrysanthemums were introduced into Europe. In Europe, the chrysanthemum signifies an expression of sympathy. Therefore, this flower was primarily used for funerals and graves. However, in the United States, chrysanthemums symbolize friendship, happiness, and well-being. Chrysanthemums belong to the Asteraceae family. Out of all the families belonging to the Asteraceae, chrysanthemums are known for being the most advanced due to their complex flower structure. The petals on chrysanthemums are called florets. Florets are small flowers that form a dense cluster known as the flower head. Disc florets are found in the center of the flower head, while ray florets are located on the perimeter. Lastly, these advanced flowers come in an array of colors, shapes, and sizes. The National Chrysanthemum Society has grouped chrysanthemums into 13 different classifications. These classifications are based on the flower's size, curvature, and overall appearance. Pesticides are substances that are meant to control pests. There are 4 types of pesticides, which are known as insecticides, herbicides, rodenticides, and fungicides. Chrysanthemum flowers contain an insecticide known as pyrethrum. The image on the screen is the chemical structure of pyrethrum. Chrysanthemum flowers contain an insecticide known as pyrethrum. Pyrethrum extracts contain 6 insecticidal esters referred to as pyrethrins. These esters act as the killing agent in the extract. To be clear, pyrethrum and pyrethrin are in fact different. The main difference between these two insecticides is that pyrethrum is the total extract from the flowers, while pyrethrins are the refined 6 esters. Pyrethrum is one of the oldest natural insecticides in use in the world today, and it has one of the best safety records of all insecticides. Pyrethrum is an effective insecticide due to its component known as pyrethrin. The table below shows the 6 compounds that comprise natural pyrethrins: pyrethrin 1 and 2, josmolin 1 and 2, and cynarin 1 and 2. The insecticidal action of pyrethrins is characterized by a rapid knockdown effect, hyperactivity, and convulsions in most insects. Pyrethrins are commonly used to control mosquitoes, fleas, flies, moths, ants, and many other pests. Likewise, pyrethrins can also control insect infestations located on the human body. Examples of these include lice and scabies. Because of their insecticide properties, pyrethrins are found in over 2,000 registered pesticide products. In general, pyrethrins are low in toxicity to people. However, pyrethrin can irritate skin and cause numbness or tingling at the site of contact. For example, if an individual is using a special shampoo for head lice and it contains pyrethrin, irritation, tearing, burns, scratches to the eye, and blurred vision can potentially occur if the shampoo gets into the eyes. When inhaled, irritation of the respiratory passages, runny nose, coughing, difficulty breathing, vomiting, and diarrhea could occur. In dogs, topical flea and tick spot-on medications often contain high concentrations of pyrethrin. Therefore, most dogs are not affected when exposed to pyrethrin. If pyrethrin poisoning is occurring in a dog, symptoms will usually develop within the first hour of ingestion. Symptoms will include excessive drooling, gagging or hacking, vomiting, anorexia, agitation, tremors, or shaking. Likewise, dermal reactions to pyrethrin for a dog can develop within 15 minutes to several hours after exposure. Symptoms will include agitation or restlessness, intense itching, vocalization, and paresthesia. However, when it comes to cats, they are extremely sensitive to pyrethrin because they are unable to metabolize these agents quickly and efficiently due to their peculiar liver metabolism. Fish are also extremely sensitive to pyrethrin and can actually die from the smallest exposure. Due to pyrethrins being easily decomposed rapidly by light, synthetic analogs known as pyrethroids were developed in the 1970s. Pyrethroids are also classified as insecticides, and they were produced to mimic the effects of the pyrethrin esters. When comparing these insecticides, pyrethroids are the most effective. Pyrethroids are synthetic chemical insecticides that act in a similar manner to pyrethrins, which are derived from the chrysanthemum flowers. In other words, pyrethroids are modified versions of pyrethrins and contain fewer chemical variants. Pyrethroids usually only contain one molecule that mimics a single natural pyrethrin ester. Due to their high insecticidal potency, generally low mammalian toxicity, and lack of environmental persistence, pyrethroids have had much success in the past 40 years. In fact, they account for about 15 to 20% of the global insecticide market. There are many examples of pyrethroids, including elethrins, cypermethrin, permethrin, resmethrin, samithrin, and tetramethrin. Pyrethroids are used widely as insecticides both in the house and in agriculture. In medicine, they are used for the topical treatment of scabies and head lice. In tropical countries, they can be used for malaria control to prevent mosquito bites. All pyrethroid insecticides contain an acid moiety, a central ester bond, and an alcohol moiety. The acid moiety contains 2 chiral carbons, therefore pyrethroids typically exist as stereoisomeric compounds. Stereoisomers are isomers that have the same composition but that differ in the orientation of those parts in space. The cis isomers are generally more toxic than the corresponding trans isomers. These chemical considerations are relevant as pyrethroids' effects on sodium channels, their insecticidal activity, and their mammalian toxicity are stereospecific. The toxicity of pyrethroids is the same as pyrethrins mentioned previously. For pyrethroids, the acute oral mammalian toxicity is generally low. Despite their extensive worldwide use, there are relatively few reports of human poisonings and only a dozen deaths. Most deaths occurred following accidental or intentional exposure to pyrethroids. The dermal toxicity of pyrethroids is also low because of limited absorption through the skin. The primary adverse effect resulting from dermal contact with pyrethroids is paresthesia. Symptoms will include continuous tingling, pricking, or even burning in severe cases. Usually in low severity instances, respiratory effects are the most common symptoms. Chronic cases with pyrethroids indicate that at higher dose levels, they can cause slight liver enlargement accompanied by some histopathological changes. An antidote is a medicine taken or given to counteract a particular poison. With that being said, there is no antidote for pyrethrin or pyrethroid poisoning. Due to its low toxicity, most patients do not show any significant symptoms. Vitamin E has been shown to be an effective treatment in dermal exposure. Immediate decontamination of the skin with soap and water can also be used to help with dermal toxicity. Optimization of the airway, breathing, and circulation is vital for any patient with acute poisoning. In conclusion, quick and effective treatment with decontamination, monitoring, and supportive care help minimize the extent and severity of signs. Based on toxic signs in rats observed after administration of 36 different pyrethroids, these compounds have been divided into 2 classes. A key structural difference between type 1 and type 2 pyrethroids is the presence only in the latter of a cyano group at the carbon of the alcohol moiety of the compound. Pyrethroids which lack a cyano group are often classified as type 1 pyrethroids, and those with it are called type 2 pyrethroids. However, certain pyrethroids do not fit into either type because they produce a combination of the 2 syndromes. When saying syndromes, I am referring to the 2 types of classifications of pyrethroids. Class 1 or type 1 is known as T syndrome, while type 2 is known as CS syndrome. The table on this slide shows signs and symptoms and examples of each type of pyrethroid. A mechanism of action describes a functional or anatomical change resulting from the exposure of a living organism to a substance. It describes these changes at a molecular level. The mechanism of action of both pyrethrins and pyrethroids are the same. The mechanism of pyrethroid toxicity is complex due to its action on several channels and proteins. Pyrethroids act mainly on sodium and chloride channels. Excitable cells are the key targets of pyrethroid toxicity. Pyrethroids are known to alter the normal function of insect nerves by modifying the kinetics of voltage-sensitive channels, which mediate the transient increase in the sodium permeability of the nerve membrane that underlies the nerve action potential. In other words, these molecules affect their host by altering the activity of the sodium ion channels of nerves. These toxins prolong the period of sodium conductance and increase the length of the depolarizing action potential, which results in repetitive nerve firing and death. Pyrethrum is the natural compound found in chrysanthemum flowers, while pyrethrins are derived from the dried and ground flowers of chrysanthemum, specifically Chrysanthemum cinefolium. In other words, pyrethrins' natural source comes from the flowering plants of the genus Chrysanthemum. However, pyrethroids are the synthetic derivatives of pyrethrin. The picture on the screen shows the Chrysanthemum cinefolium. In conclusion, chrysanthemum flowers contain a toxic component within their unique flower head. From this, humans have manipulated this natural toxin to create insecticides that can be used by all humankind. Although this toxin can be harmful to mammals, the benefits definitely outweigh the overall risk of toxicity. With that being said, the risk of severe or chronic toxicity is very low and can even be considered rare.